So if you are like most students, this concept is kind of a nightmare. You look at the diagrams in the textbook and you're saying, well, I can sort of understand that this is a mitochondrion. And now that when I'm looking at the detail, there are just like, there's a whole bunch of stuff going on. There's lots of bits and pieces. You've got the molecules that are here. You've got this gigantic thing, the ATP synthase. You've got the movement of electrons. You've got the movement of hydrogen ions or protons. How do I keep track of this? Okay. It's really hard if you approach it the wrong way. I mean, if you take the approach of trying to, get the details lined up and make some sense out of the details, that's kind of the wrong way to do things. Okay? And I'm hoping that with this lecture, with this video lecture, I'll be able to give you that other perspective, thinking about what's actually going on first. And once you get that, the whole concept of how oxidative phosphorylation is going to be generating the ATP is going to make a lot more sense. And that's as far as I want to take you, right? Later on, when you take physiology or some other class that is, maybe you'll become a biology major and you'll take BIO110. And the actual dynamics of what's going on at each step in electron transport, okay, will be more important, and you'll get that detailed view later on, right? Right now, I just want to give you the big picture with an adequate level of understanding, because it is, even so, I mean, even though I'm giving you the big picture, it's still going to be a little bit complicated, and it has to come after you understand what's gone through in the process of glycolysis and the citric acid cycle. So let me return to, actually go to the whiteboard for the first time here. And we're looking at now oxidative phosphorylation. And maybe I should preface this by reminding you of things that are told to you in the textbook, right? Now, one thing you know from the textbook is that we're accounting for the majority of ATP production, right? 32 out of 36 ATPs that are produced in the cellular respiration, the metabolic consumption of glucose with oxygen, right? We only get 4 ATPs between glycolysis and the citric acid cycle. The other 32 ATPs are going to have to come from someplace else, and that someplace else is oxidative phosphorylation, right? So yeah, it's important. This is where we're getting all of the ATPs being produced, okay? The things that we've already talked about, glycolysis and citric acid cycle, they're producing a little bit of ATP, but they're really setting the stage for oxidative phosphorylation, or as we sometimes like to say in biology speak, ox-phos, right? So you could say glycolysis and the citric acid cycle, they set the stage for ox-phos. And how do they do that? Well, they provide the electron carriers, right? And so, yeah, the two things that are needed, the two consumables that are needed by oxidative phosphorylation are one, the electron carriers, NADH in particular. NADH is the one that we highlighted in our previous discussion when we said that glucose is oxidized. The electrons from, actually 2 electrons from glucose are being put onto NAD. Actually, it's NAD plus. And in the outcome, the product that we have after adding the 2 electrons, we're also adding a hydrogen ion, is NADH, right? And so we went through lots of examples of this in glycolysis and citric acid cycle. Now we have, I think, 10 molecules of NADH, 2 molecules of FADH2, and these are ready to go into the electron transport system, which is the first part of ox-phos. The other consumable that's required, and this is really where oxygen is coming in. The other consumable is oxygen, right? You might go back to the previous video, and you'll note that even though we know that oxygen is consumed in cellular respiration through glycolysis and through the citric acid cycle, there actually is no oxygen that comes in, right? All the oxygen is coming in at the end of the electron transport chain. That's also where the water is being produced, right? Because water is what you get after you add the electrons to oxygen, okay? Now, the other thing that we need, I mean, it's not simply enough to have the electron carriers and oxygen. We need to be able to have the infrastructure, the cellular infrastructure, they call it the machinery, which is basically part of the structure of the mitochondrion. And what I want to go over with you is the idea that mitochondrion is a literal battery, literal meaning not metaphorically, right? And so in the same way that a battery is charged and discharged by charging, we're basically putting a whole lot of charges on one end and negative charges on the other end. And yeah, if we put a wire across here, we could actually have the negative charges, the electrons flow over to the positive side, and that's going to give rise to discharging the battery, but that discharging can be used to drive, to power something, to power your iPhone, to power your alarm clock, to power your whatever it is, a flashlight. Okay, and here's one of those kind of rare situations where we're not really using a metaphorical thing from our normal consciousness to speak about a biological concept. A mitochondrion actually is a battery because we are charging a mitochondrion with a positive side and a negative side, okay? And we're also discharging, okay? And the discharging is a part that's going to be, in the same way that the battery is discharging, is going to be used to power the lighting of a flashlight or keeping your iPhone from working. In this case, the mitochondrial discharging is going to be associated with ATP synthesis, right? The charging is going to be the thing that is going to be involving the moving of electrons from NADH to the ultimate electron acceptor, oxygen. And so electron transport and ATP synthesis are coming in as kind of like the outcomes of having a mitochondria that's doing its actual work. Okay. And so on the next slide here, I've got a diagrammatic picture of the mitochondrion showing the outer membrane and the inner membrane, outer, inner. Right. And the two compartments that we have here are the inner matrix, and the intermembrane space. Okay, so I didn't draw the inner membrane all convoluted and such, which like the way that it's drawn more accurately in the textbook. All I want you to have is the idea that we've got two compartments, the inner matrix and the intermembrane space, right? And so what I want you to visualize here is we've got two processes. I'll kind of diagrammatically divide the mitochondrion into two halves. The top half will be charging mitochondrion, and by charging the mitochondrion, we'll be taking hydrogen ions from the inner matrix and moving them into the intermembrane space, H+, 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 right? And consequently, we're going to be making a high concentration of hydrogen ions in the intermembrane space, low hydrogen ions in the inner matrix. This is basically an example of active transport. Only the mechanism of moving these hydrogen ions is going to be the electron transport system. Right? Discharging is taking advantage of the fact that we've got a high concentration of hydrogen ions in the intermembrane space, and we're going to be allowing them to move into the inner matrix because we've got a high hydrogen ion concentration in the intermembrane space, a low hydrogen ion concentration in the inner matrix, right? And so, yeah, and so far we're kind of talking about a concentration gradient. Yeah, there will be a high concentration of hydrogen ions on this side in the intermembrane space. Okay? We'll have a low concentration in the inner matrix, and that's going to be setting up the one way of thinking about the battery. But I said this is a literal battery because we actually have a positive charge because hydrogen ions carry a positive charge. Having a low concentration of hydrogen ions in the inner matrix means that there's going to be a negative side and a positive side. So we actually do have a electrochemical gradient, right? There's electricity as well as an osmotic or a diffusion gradient between these two things. In other words, if you went in with a little electrical meter and put one probe on the inner matrix side, another probe on the inner membrane side, you would see a real voltage. Okay? There's a voltage difference between the two, and that's what makes mitochondrion a literal and not just a metaphorical battery. Okay? Okay. So the idea here is that we've got two things going on, a charging and a discharging. Okay? And as long as we have a continual metabolism, the charging is going to be taking place at the same time the discharging is taking place. And that's the first part that we want to make sure that we understand, right? So yes, charging of this mitochondrial battery involves taking the hydrogen ions, the positive charges, and moving them into the intermembrane space, right? Away from the inner matrix. Okay? At the same time that that's happening, we're allowing some of the hydrogen ions to come back into the inner matrix, and that constitutes a discharging of my battery, right? Now, the other side of this is that the discharging of the battery is going to be coupled with ATP synthesis, okay? And the charging is going to be occurring as a result of electron transport, right? Remember, the two things we need to account for in our understanding of chemiosmosis, oxidative phosphorylation, is electron transport. We have to be consuming NADH, FADH2, and oxygen, right? That's going to be happening in electron transport. Electrons are taken off of NADH. We're ending up with NAD+. The electrons, two of them, are going to go to oxygen. Half a molecule of oxygen is going to be giving rise to 1 molecule of H2O, right? So this is basically what's going on in electron transport. And this is also what pushes the hydrogen ions from the inner matrix to the intermembrane space. Okay. So let's go back to our battery concept. Okay, the two things we need to account for is electron transport right? And these are the molecules, and the molecules we saw in that scary diagram, right, are the molecules of the electron transport system. Electron transport, right? And so electron transport is doing exactly this. Electron transport system is taking up NADH, releasing NAD+. We're getting the electrons away from NADH. Electron transport is taking up oxygen. Electron transport is reducing the oxygen, producing H2O. Remember, H2O is kind of like the end point. That's where the electrons are at their most stable position, right? Now, because energy is released as electrons are moved towards places where they're more stable, that energy released is realized in the form of the H plus movement, the H plus being actively moved from the inner matrix to the inner membrane space. Okay? And so electron transport means that this is a mechanism that we use to charge the battery. Okay? The other side of this, the other thing that we need to account for is ATP synthesis. Okay? And we know that ATP synthesis is going to be requiring a lot of energy. And here the energy is coming from the battery. Okay? As hydrogen ions are allowed through, we've got another structure, this ATP synthase. that take the energy that take advantage of the energy that's stored with the high concentration of hydrogen ions on the positively charged intermembrane space side of the membrane, allowing those H plus to move into the negatively charged low hydrogen ion concentration inner matrix. This is the part that's going to be generating ATP from ADP and phosphate. Okay. ATP synthesis, ADP and phosphate, in, out, ATP. This is going to be driven by the energy potential that comes from the high concentration in the intermembrane space to low hydrogen ion concentration in the inner matrix. Okay, so two things going on, keep them straight. Charging, discharging. Discharging, ATP synthesis. Charging, electron transport. Okay, think about that for a little bit. And then you could return back to that picture that we had before, this one, and maybe it'll make more sense now. Okay, remember, all this here, all this here is electron transport. Okay, and you might notice that electron transport is taking electrons from NADH, okay, and ultimately putting those electrons onto oxygen. Right, the electrons are just flowing from molecule to molecule, but ultimately those electrons are put onto oxygen, and we end up with water as the end product. Right, that's what we just said. The other thing that's happening as a result of electron transport is we're seeing the movement of hydrogen ions across the inner membrane to get to the intermembrane space, right? And we're doing this quite a bit in the case of electron transport. It's the electron transport side, right? This is the charging. The other side is the discharging. And as protons are allowed to move through into the inner matrix, that's basically turning a little turnstile, which is driving ATP synthesis. ADP and phosphate is using the energy of that movement of hydrogen ions back into the inner matrix to generate ATP. And this is where the ATP is produced in oxphos. Okay. And these ATP join all the other ATPs that are produced in glycolysis in the citric acid cycle, and they're consumed by cellular work that's going on in the cell, which basically restores, brings us back to ADP and phosphate, which have to come back to cellular respiration mechanisms to generate more ATP, consuming more food, consuming more oxygen, and the cycle goes on and on and on and on, continually 24/7 to the tune of a huge boatload of ATP being produced every 24 hours. Okay, now this, I was a little bit hesitant to do this, but I decided that this is actually a really nice exercise for us, which is to consider what's going on in the case of brown fat. There's a weird little chapter that comes after this one, talking about brown fat and the fact that we thought brown fat was only active in infants and very young children. We thought that by the time you become an adult, the brown fat is completely gone. We're interested in brown fat because, well, if you were able to activate brown fat, you'd be able to burn off all the excess calories. You could eat as much as you want. You could totally pig out on the most caloric foods that you want, and you wouldn't have to worry about gaining weight because you could have your brown fat just convert all those calories into heat, right? So the idea is that brown fat converts food energy into heat, right? And therefore, because we're taking the food energy and converting it into heat, there's nothing that would cause us to actually have an excess of energy, excess of energetic molecules to be stored in the form of fatty tissue, right? Sounds wonderful. I struggle with my weight, and so I think it would be really awesome to basically say, okay, well, yeah, I think I overdid it at that last meal. I think I'm going to just turn my brown fat on a little bit and burn off those extra 1,500 calories that I should not have eaten, right? That would be pretty awesome. Okay. What does this have to do with mitochondria? Well, I'll tell you, right? Because if we were to take this portion here, right? Remember, this is the discharging. Okay. If we were to discharge, if you were to take this out, remove. And if you're to allow your mitochondrial battery to discharge without generating ATP, okay. In other words, if we're just like open a channel, okay, this ATP synthase harnesses the energy as the protons are passing back into the inner matrix. If I said protons, hydrogen ions are protons. If these hydrogen ions were allowed back into the inner matrix for free without generating ATP, then in effect, we would be consuming all the food energy, but we wouldn't be producing ATP, right? And so if you think about that, we've got a nice little chemical equation going on here. We know that we're taking food molecules and oxygen. We're still producing waste molecules. And that's just going to be CO2 and H2O. In other words, the same amount of energy is being produced here that normally generates 36 molecules of ATP, right? So in other words, if we normally would be producing 34 molecules of ATP during oxidative phosphorylation, as well as some heat. Remember, because heat is always being produced. What we're saying here is that if we prevent ourselves from making all this ATP, if instead of producing 34 molecules of ATP, we produce fewer. Let's say we don't produce any. We cut off ATP production altogether. And so there's no ATP production, but we still have that same energy coming off. That energy has to be going someplace, right? And so in the case of a mitochondrion that is not producing ATP, all that energy is going to be realized in the form of heat, right? In other words, they'll be producing a lot more heat. And this is how we can have the generation of heat with brown fat. So this is the extra heat. Brown fat, which is basically a whole lot like regular fat, regular yellow, creamy colored fat, except for a couple of things. One, it's got these faulty mitochondria. Mitochondria are not very effective. They've got their low efficiency mitochondria, and so they don't produce a lot of ATP energy, but they do consume lots of food. They're also brown because they've got the pigments to take up lots of oxygen, right? So taking up oxygen, generating a lot of heat. And this this is why they're really, the reason why they're really good for infants is, well, babies tend to be small in size and they don't have a lot of body fat. They don't have a lot of hair covering, so they tend to lose heat very rapidly. But one thing that they don't lack is a calorie source, because baby mammals have, they nurse, they take in mother's milk, and they're basically fed a high calorie diet. And yeah, a lot of those calories can be consumed, can be dedicated toward the production of a little bit extra body heat to compensate for the fact that little babies tend to be small and relatively hairless. Okay, and so that's the context of what you might be reading in that next portion of the textbook. Okay. Other than that, I think we're pretty much done with today's discussion. And as I said, what I hope that you're able to do now is maybe go back to that chapter with chapter 6, part 11, where you've got that, that challenging picture of oxidative phosphorylation, electron transport, and ATP synthesis. And I'm hoping it'll just make a whole lot of sense to you now, and you won't feel so intimidated the next time you come across this in your biology class, in physiology, for example.